I am in the cytoplasm on the ribosome. The ribosome aligns the messenger RNA in a way, making it exposed for those tRNA molecule. And as we said earlier, every tRNA comes with two parts. You have the amino acid part, where the amino acid is attached, and you have this triplet at the bottom that we call the anticodon, right? So the first triplet on the messenger RNA is what we call the start triplet, right? You have to remember that start triplet because it's always the same. We call it the AUG triplet. So that's always the first triplet on the messenger RNA. Now, AUG would correspond to what? It would pair up always with UAC. So that means that I'm going to put first amino acid here at the beginning. And then I have another triplet here, right? The codon, the second codon is called UGU. You can see it here. So now this will call for the second tRNA and the second amino acid. So there we go. So I have ACA that will pair up with the other triplet. So as you can see, what am I going to do here? I am basically putting one amino acid after another. And every time I have like two amino acid adjacent, like this, what's going to happen? The ribosome will catalyze a reaction, condensation reaction again, same as the one we saw earlier today. And that condensation reaction will form a bond between amino acid. We call that bond between amino acid, we call it a peptide bond. So that's we're forming a peptide bond through a condensation reaction. All right. So now that we've joined the first two amino acids, we're going to do more of this. So we're just going to bring some other tRNAs, some other amino acids. And as you can see, the polypeptide chain is getting longer. So those tRNA will soon exit. They will no longer need it. And you will be left with what? You'll be left with a polypeptide chain. So